What am I most excited about? Bespoke CRISPR therapies.
Dr. Jennifer Doudna is a Nobel Prize laureate and CRISPR pioneer turning gene editing into real cures.
A child treated with a therapy that was created for him.
We talk about her efforts to help treat a child with a devastating genetic disease.
This is probably not a one-off case.
And new ways to help improve health by working with the microbes that live in and around us.
We're in a moment where science is really under attack. Science communication is more important than ever.
What do we do?
Welcome to Stanford Department of Medicine's Inside Look at the Future of Medicine.
Well, Jennifer, welcome to Stanford.
Thank you. Real pleasure to be here.
Yeah, thanks so much for coming down the coast to visit us.
Easy ride.
Yes, pretty good. I was preparing for the conversation with you today, and I realized that it was only about 10 years ago, just a little bit over 10 years, that your paper in 2012 in Science that really kicked off the CRISPR revolution. For some reason in my head, so much has happened. It seemed like it must be much longer, but 2012. In that decade, I mean, so much has happened. Your life has changed a little bit in a few ways. What has surprised or delighted you the most in the last kind of 10 years or so since that?
So many things have happened, as you said, and some of it has been on the science front, and some of it's been on the, frankly, larger scale of public policy and where medicine is headed, where agriculture is headed with these kinds of tools now that we have for manipulating DNA. It's really been very interesting to see all of that unfolding. What am I most excited about? I guess certainly top of mind today is the opportunity to create bespoke CRISPR therapies for patients in real time. I think this is such an interesting moment we're in right now. We've seen the example with baby KJ. We talk about that right here today. Yeah. And there are so many things going on here at Stanford and in the Bay Area, and of course much more broadly across the country and elsewhere, that make us think that, there really is an opportunity right now to seize the moment, figure out how we can find those patients that can benefit from CRISPR and create the pathway to, helping them in real time and doing it in a cost-effective way. That's what I'm
Scaling
really
the thing.
Really interested in doing. Yeah.
Well, we'll get to a little bit more detail on the baby KJ story shortly, but you lead an institute with, I think, one of the coolest names of any institute, the Innovative Genomics Institute. As a genomics nerd, that sounds like the perfect place for me to visit or work. I think who wouldn't want to work there? But tell me a little bit about the institute first and the work that you're doing there, and then we'd love to dive into some specific examples.
Well, this institute started just over 10 years ago. Okay. In Berkeley and in San Francisco. We were a joint institute from the very beginning, linking UCSF and UC Berkeley and UC San Francisco, with the idea that we could bring genome editing to bear on health and climate challenges in ways that would have real-world impact. How do we do that? It sounds like a broad, grand goal, but how do you actually get there? And what I'm excited about is that over the 10 years of our existence, we've raised a lot of money. We've also invited a lot of partners in. We've just recently signed an agreement with our third campus partner, UC Davis, has become a formal partner of the IGI. And we love the idea that we can leverage expertise that exists on different campuses to focus on big projects and problems that none of us individually would be able to work on or really tackle meaningfully. And what's amazing is that we're really focused now on building collaborative teams, especially with younger scientists who are just starting their careers, and with companies that can bring their expertise to bear on these projects and problems, and inviting people that are very excited about science, but maybe you're not scientists, philanthropy, it comes to mind, that can meaningfully gather their different capabilities to do things that otherwise wouldn't happen.
So you view the institute as partly as a sort of gathering place, bring the smartest people, the most energetic people, the most innovative people together and help them do cool.
I mean, that's what we do here in California, right?
That's what we do. The new frontier. Yeah, I think that's right. Perfect place to do it.
Yeah.
I'm really excited to be able to collaborate on a few of these things. And
absolutely.
I love many things, but what I love about it most is these ambitions are not small. Like human health and disease and planetary health are pretty big topics. I don't think anyone would doubt that you could make an impact on those.
Why don't we start maybe with this amazing story that hit the headlines in this last year with the collaboration that you helped put together and the teams that came together for Baby KG. How did that come about? And tell us how things are going.
It's an incredible story, isn't it? It's so inspiring to see the example of a child who was affected by a metabolic disease, diagnosed in real time, and treated with a therapy that was created for him, for his disease, and tested first in animals and in collaboration with appropriate regulatory guidelines and his clinical team at the Children's Hospital of Philadelphia, figuring out how to actually treat this patient and ensure that he would have a safe path to what we hope will be a permanent cure for the disease. It's really, really exciting. It's worth pointing out that this is probably not a one-off case. This is an example of what is now possible to do. And it's partly, it's inspiring to see a patient and his family, of course, is, treated effectively. But what's even bigger, and I think, inspiring to all of us, is the opportunity to do that for so many more.
And the key features of that, this was CPS1 deficiency. So this was a metabolic disease that causes hyperammonemia, and that can cause severe brain damage and would even be associated with a 30 to 50% risk of death. So this is not a trivial condition, and one that was diagnosed pretty early in life, like 2 or 3 months when he was first diagnosed.
Yeah, I think even earlier.
Yeah. And then so many remarkable things about it. I mean, maybe in your world, the least remarkable thing is that you can edit the genome to a better copy. But being able to take that case and with the joint work with the group at CHOP and at Penn to be able to develop this, test it, go through the regulatory agencies, and within, I think, was it 7 months from? Yeah. And then to have an N of 1 therapy that was essentially curative. I understand there were 2 or 3 doses that were given.
That's right, 3, yeah.
But this is the ultimate in many ways. I think that's one of the reasons they caught people's imagination in precision medicine. Here is a precision surgery for the genome that is essentially curative of a disease that would otherwise be fatal. I mean, that's incredible.
It's incredible. Yeah, it's really very exciting. And as you're saying, I think the inspiration is part the effect on this individual, but it's also the idea that we could do this for many others. And we now have a pathway. We can see all of the steps that can happen and how to do them in a period of time that's meaningful for the patient. It doesn't take years and years. This was done in a matter of months.
Right.
And wouldn't that be great to do for more?
I mean, there are thousands of these babies born every day. And I think being able then to scale. And so you can see a path towards not N of 1 just, but N of 10, N of 100, N of 1,000. Yeah. And I think one of the challenges has been that technology often moves so much faster than our regulatory agencies are able to. I mean, they have a very important job, that safety is incredibly important. It's hard when there's so few people in the world who truly understand new technology. But it seems like there was a very collaborative relationship with the FDA over this, and that they are very open to the idea of scaling.
That's what we're seeing. It's really interesting to see this. And I think it's partly, again, that, the FDA, after all, is human beings, and they can see the potential. They can see the opportunity. I think, Fyodor Urnov at the IGI deserves a lot of credit for his role in helping to educate regulators about the science. As you said, it's often hard for them to keep up. There's so many things happening. How do they become experts in every new technology? They really can't. And so it does require scientists to go in and say, look, we'd like you to understand what we're doing. We want to work with you. We want to partner with you. We want to do this in a way that makes sense for everybody. And of course, safety has to come first.
Yeah. It's great that it's a conversation, because I think once you're conversing, it's possible to have those explanatory moments. And I think, so there was an industry partner as well. I think this was in this large group.
That's right.
Danaher were involved.
Yeah, it's very interesting because that partnership existed already, was kind of pre-existing to the diagnosis of KJ. This is a partnership with a company called Danaher that makes molecules, that, among other things, and they are very good at it. They know how to make molecules that are prepared in a way that can be delivered clinically. And so when the baby KJ case came along, they stepped up and said, yes, we're willing to provide the molecules needed for this treatment, and we'll do it at our expense.
Yeah, amazing. Yeah. And then this was a base editor delivered in a lipid nanoparticle, but so
right,
it goes to the liver pretty naturally, which is exactly where you need the.
Well, you're touching on an important point that I think is really good to just state here very clearly, and that is that one of the reasons that the baby KJ case could proceed the way it did was that the affected tissue, the liver, is one that we know how to deliver molecules to. We used existing technologies with both CRISPR and the delivery vehicle, the lipid nanoparticle, but were also pre-existing, that had been clearly validated clinically. And so there was no new technology that needed to be created. We simply had to refashion it for this particular case, but we know how to do that after 10 years or plus of doing this. Yeah. And so I think that, right there tells you that, when you have technology that's reached a place where you have off-the-shelf capabilities, we need to get better at quickly, pointing that to the problems where it can really be used in real time. And that's what was so exciting here.
Yeah. And it sounds like there's openness on behalf of the regulatory bodies to see that many of those elements can be repeated, and maybe just the guide RNA can be changed, and you have a new therapy for a new patient.
And this is game-changing, isn't it? I mean, you know better than any of us know, thinking about when you have rare genetic diseases, this is something that, in the past, we would have said, well, we can diagnose it, we can maybe give palliative care, but we really don't have anything that's going to provide a long-term or even potentially a curative.
Yeah. If the best outcome is a liver transplant, then hopefully we can do better than that.
Exactly. And this is what CRISPR has offered from the very beginning, right? Is this opportunity to create a targeted approach for each individual mutation. It's just that, if you have a rare disease where for each disease that you're treating, you have to go through a full-blown 3-phase clinical trial, it's not realistic, right? I mean, you don't even have the patience to do it, even if you wanted to, and the expense would be, prohibitive. But here, I think the FDA is saying, look, let's take a new lens to this. Let's recognize that this is really a different kind of technology, and we're really talking about a different kind of therapy, aren't we? This is not something that you're going to be injecting someone with every day or a month. Hopefully it's a one and done.
One and done. Yeah, remarkable. Really, it's the sort of ultimate for precision therapy. Yeah. And amazing that we're able to talk about that now. We made science to cure. Now we just need to think about scaling.
Exactly.
So, well, talking about scaling, that's talking about an end of one therapy. But you also have these ideas to potentially impact the entire population, or thinking about the planet, by editing the microbiome. And I realize that you recently got a big grant with Joe Banfield to think about microbiome editing. I spent a lot of time thinking about genetics, genomics, and also gene editing. I hadn't spent a lot of time to date thinking about microbiome editing. So tell me what's in scope for that. Sounds really exciting.
Well, what's fun about microbiome editing is it takes CRISPR back to its source. Yeah. Right? So maybe a lot of people know this, but maybe they don't. The CRISPR comes from bacteria. It comes from an immune system that bacteria evolved to fight viruses. So we think it's very interesting to now take that technology and turn it back to those very microbes and use it in a way that allows targeted changes to be made to particular microbes in the context of a multi-species community, which is what a microbiome really is, right? And doing that in a way that could impact health and could impact, well, let's say just health writ large.
Yeah.
Health of humans, health of the planet.
Right.
And so that was the vision for the program that we call BION, which is our TED Audacious funded project with Joe Banfield. Yeah. And the idea there was to create the kinds of targeted tools that will allow CRISPR to work in the microbiome, whether we're talking about the human microbiome, where the cow rumen
Yeah, I was going to ask,
what the other
what sort of example
when you were working.
could you get? So, but potential climate change is on the agenda here.
It's on the agenda because there were, and this, involves our wonderful colleagues up at UC Davis who have for a long time been aware that the microbiome in the cow rumen, which is where, they're digesting the grass and multiple stomachs and got a lot of microbes that are busily doing that kind of metabolism. The problem is that some of those bugs are also making a lot of methane.
Yeah,
And that's one of the most powerful greenhouse gases. And it's
like a third or something of human produced.
shocking, right? I was astounded to learn that agriculture, and in particular cattle farming, accounts for almost a third of the methane emitted annually.
Yeah.
That's, sort of human caused. So what can we do about that? I don't think it's realistic to ask everybody to stop cattle farming,
Yeah,
right? That's just not going to happen. And so better would be to find a way to actually just reduce the methane emissions at the source. And so this is where we think CRISPR comes in, because we know we can manipulate genes in these microbes.
right.
Increasingly, we know the genetics of methane production in the cow rumen. And so again, it's sort of analogous to the baby KJ case where we've got all the pieces.
Right.
We've just got to put them together to make something that's going to have real impact. And that's what we're doing with our biome program. So we have a whole team of young investigators. Jill Banfield is kind of the leader, but we've got a lot of scientists now who are really focused on this kind of challenge, working both at the Berkeley site of IGI, but also working up at UC Davis, where they actually are birthing calves and starting to test.
Right.
So it's great.
The idea is that we would treat individual cows.
Individual cows, and again, the idea would be a one and done
Yeah.
kind of treatment that would then be maintained through adjustments that wouldn't be prohibitively expensive in the diet
Yeah.
and would be possible to distribute around the world.
I see. Really interesting. And then I'm guessing, I'm just thinking this through now as you're explaining it, but I suppose offspring often, they don't inherit the microbiome, but they are gifted much of their microbiome from their parents at the time they're born. Their mother predominantly at the time they're born, but then I assume in an animal population that there may be some chance that some treatment of the mother might even impact.
It's an interesting question, right? I mean, that's one of the things we're researching right now, is to what extent, do you get that kind of
population
change?
Yeah, that's really, really fascinating. And really, CRISPR writ very large indeed. I mean, this is literally a climate level question. There's some human health elements to that too. Did I see asthma maybe as one disease that you think?
That's right. So Sue Lynch up at UCSF, you may know her. She has had a long-standing clinical research program on the connection between the human microbiome and asthma.
Yeah.
And so we would love to be able to also manipulate the human microbiome to reduce asthma susceptibility. Sounds a bit wild. I thought it was it sounded a bit like a long shot when we first discussed it. But I think the pieces are coming together. We understand the genetics of production of molecules in the human gut that can induce asthma susceptibility. And now, we have the tools to manipulate it. So you can start to see the pieces coming together.
I think in many ways the microbiome has been really ignored by human physicians and by the healthcare system to date, partly because we haven't been able to manipulate it very well. Obviously, C. diff diarrhea has been one area of great success, but overall, I think we've found it fascinating from a scientific standpoint as we learn more and more ways that the microbiome interacts with the human part of the dual organism, and vice versa. Obviously, a lot of diet and nutrition work and our GI colleagues have been to the fore. But some of the remarkable impacts that are seen even on psychological conditions and psychiatric conditions, on anxiety from microbiome is just remarkable. And I do think that we're ready for another revolution there, where we understand that we can actually manipulate the microbiome in a much more precise way to help with these diseases where it's clearly fundamental. Even responses to chemotherapy and other trials that we've seen by the microbiome could be significant.
I think the science looks so interesting. It's not my area of expertise at all, but I'm fascinated by the increasing, as you said, connections with different human disease conditions. There's a lot of fundamental science still to be done, but now we have the tools to do it.
we have a lot of people you're here talking to are trainees and many others here at Stanford, and we have many who have thought, including in our faculty, who have thought about moving into industry. Some actually, this be West Coast, come back from industry back to academia. We have pretty free flow, and we have many who start companies as well. But I wondered, since we have you here, you're both somebody who spent a short time, I believe, in the industry, but also
Very short.
yeah.
Yeah.
And maybe not everyone knows that story. If you want to share, I'd love to hear that. But also, you've started companies, obviously, and moved technology really into the real world. Do you have advice for people that are thinking about that or who are struggling with that element of what the next career move?
Well, it's a great question, because I consider myself the most naive person probably out there when it comes to thinking about the how to sort of commercialize scientific discoveries. I certainly didn't. That was not on my radar when I started my career, and even when we started working on CRISPR, it wasn't the thing that was on my mind at all.
Well,
And I had to learn about it, because I realized fairly quickly, once we had done the initial work on CRISPR, that when you have a technology that's cross-cutting like this, and you see opportunities for it to have real impact, some of that can get done in academia, but frankly, you're probably going to need the teams and the financing to do it much more broadly than would be possible in a typical academic laboratory. And so how to do that became top of mind for me at that point. And I was fortunate to have a lot of people who, including a number of folks at Stanford. I consider, by the way, I've long considered Stanford a real leader in this. Stanford has had a culture for a long time about how to make that smooth transition between fundamental discoveries and scaling that happens with commercialization, and how to do that effectively. It's been a great journey. I can just give a 30-second summary, because you alluded to my little foray into the business world. And this happened in 2009. So I had really exciting opportunity to move to Genentech. And I decided to do it because I felt that I was sort of, mid-career at that point. And I'd been at Yale for several years before I moved to UC Berkeley. So I'd been at two wonderful universities. I was starting to worry a little bit that my work, although exciting on the one hand, was not going to have the kind of impact especially in health, that I would always write about in my NIH grants, and I thought, am I really
turns out you were wrong about that, but yes.
Well, but this is part of the story, right? Because I accepted the position at Genentech, and it was a bit of a tumultuous time there. They were being purchased by Roche at the time, and so there were lots of changes happening. And I just realized within a few weeks that it was, a little bit like putting a square peg in a round hole or something. I do love the academic setting in the sense that I love the lab. I love working with my students. Yeah. I really enjoy that interchange. So I ended up going back to Berkeley. I had taken a leave. Fortunately, it took me back.
It took you back, okay.
But an interesting thing happened, because when I went back, I'd cleared my calendar completely, as you can imagine. I didn't have any travel.
You weren't going to be there.
And I didn't have any teachings, And so I suddenly had, I looked at my calendar and I had like these blank days of just being able to hang out in my lab, talk to my students, think about things, work on things that I just found interesting. One of them was CRISPR, because this was something we were playing around with due to Jill Banfield, right? Her ideas around what it might be doing. And because of that, in part, I think it really gave us the freedom to explore some ideas that might or might not have happened otherwise. And so I really taught myself something interesting in that whole process, and that is that, I think it is important to pursue your scientific interests. I think it's good to keep an eye towards applications and thinking, where could this discovery have impact? But I do think it's important to be true to what you really love to do, and that's kind of what I taught myself in that experience.
That's remarkable, actually. What the journey, the path not traveled,
Yeah.
But I think this idea that you need space to be able to think and explore, and how important it is to explore without understanding exactly where the destination would be.
Yeah.
is
It's really valuable.
is
And
amazing.
it's something we all love in our
Yes.
academic
Yes.
labs. Right?
Maybe I should clear my calendar for the next 2 weeks. You've inspired me. Well, talking about inspiration, I know that some of your early inspiration towards science came from reading books, and that was certainly true for me. Many people, I think, have very inspiring teachers in school. That probably wasn't the case for me in science, but one of my teachers did give me a book, The Selfish Gene by Richard Dawkins, that
Oh, yeah.
really led.
Yeah.
I mean, and just his writing was so clear, and it just the passion for the subject was just came across, and it really lit the flame for me. But you're really, I think, viewed of course as one of like the leading science communicators in the world. And we're in a moment, kind of interesting moment, where science is really under attack in many ways, and certainly science communication is more important than ever. And I just wondered if you had some thoughts to share about this moment we're in, how important it is for scientists like ourselves to be out there talking to the real people in the world, not just the folks in our lab, but the folks at our conferences, as science is somewhat under attack.
And it is a fascinating moment because on the one hand, we've got more tools than ever for communication, right? We got all kinds of social media.
Yeah.
We've got many people who have stepped forward as spokespeople for science, which is great.
Yeah.
And a lot of those folks have many followers, and they've attracted a lot of attention and a big audience on the one hand. And then on the other hand, there's a lot of, frankly, just false information and disinformation that goes around. Vaccines come to mind, for example, where unfortunately it does a tremendous disservice to the science and also to people that could otherwise benefit from it. So what do we do? And I think that it's a tough challenge that I'm not sure there's an easy answer to. I do think that scientists need to be better about communicating about their work. I don't mean that all of us need to become national or international lecturers on the topic. It's just that I think all of us need to figure out where can we have an impact. It's as simple as having a 1 or 2 sentence non-jargony explanation of what we're doing, so that when you sit down on a plane and you're chatting with your seatmate about what you do, you can roll that out. Get an elevator, you can tell somebody quickly what you're doing and why it matters. Unfortunately, because scientists haven't been effectively doing that over the last few decades, there's been a bit of an erosion of the appreciation of science across our country, and I think some taxpayers at least wonder, why are we shelling out money for projects on things like bacterial immune systems, and why does that matter, right? And so I think it is very important to do this. So this is what I tell my students. I said, figure out where you're comfortable. You don't have to, you don't have to be an author. You don't have to be a lecturer necessarily unless you want to do that. But just being able to tell your grandmother, what you're doing and why it matters is really key.
I think that's something we talk about a lot here, actually, and I think it's an example we often use is like your Uber driver, when you're around the country giving a talk or something, and you're often talking to your
Exactly.
It's those conversations I think can really have an impact on people. And like you say, just being able to understand, I often say this to my students and trainees when they're giving a scientific presentation, you actually want to think about it as if you're explaining it to your grandmother or explaining it to a family member, because that will force you into a narrative format to sort of tell a story.
Exactly.
And even scientists like to hear stories.
They do.
They want to see your data, but they love to hear stories.
That's right.
And I think that we could all be better at that. And I think that in order to gain a little bit of the public's trust, and we have collectively lost some of that, whether it's because of folks who are treating science as something other than the rigorous approach to truth that we know it to be, or whether it's because we're less good at communication, we definitely have lost a step. And so either way, I think we all do need to step up a little bit.
Agreed. Yeah.
I wanted, while I had you captive here, to ask a specific question that there aren't many people I could ask this to, but I was at this you did some listeners may not know, but you did your PhD with Jack Szostak at Harvard, focused on ribozymes, catalytic RNAs, and studying essentially the origins of life.
Yeah.
And I happened to be at a conference recently, and Nobel Laureate from Arizona was there, and he had run this mission, the OSIRIS-REx mission, where they sent a capsule
to Bennu, which is this 4.5 billion year old asteroid, in order to capture essentially space dust that was unimpacted by human, non-contaminated by humans. And it just kind of blew my mind that they found on this asteroid that's 4.5 billion years old all the nucleobases and 14 of the proteinogenic amino acids and many of the things that would be required for the origin of life. And this is something that where you started your career, and I sort of have a hobby interest in it just to read about that. But the RNA world idea is one of the ones. I think that's in many ways what drew you into RNA. I wonder what you thought of that. They just have been publishing in the last few months these papers with this incredible like prebiotic chemistry analysis, and you must be an RNA world person, right? That do you believe that?
Whoa.
Right.
Yes.
It just seems like, I mean, that's one of I mean, we've talked about individual humans, we've talked about planets. This is always like, you know, planetary scale ideas, like thinking about how life could actually have started.
It's such a fascinating question.
Yeah.
Right? I mean, it's just so interesting. When I got to graduate school, I really didn't know what I wanted to work on. And my advisor, Jack Shostak, was brilliant at taking a huge question like that
Yeah.
and boiling it down to experiments that we students could actually do in the lab. You know, it was really interesting to me to see how you do that, how you think about, and we try to do this, of course, in our own research, right? As we have, you know, big questions we want to answer, but you've got to somehow figure out how to turn it into a, you know, an experiment that somebody can actually do in a tube, or, you know. And so that was my first real exposure to that type of science, and I've never lost that sense of wonder at, you know, the kinds of, you know, capabilities that scientists have when they really put themselves to a task like that. And, you know, in the case of the RNA world, yeah, I think it's still a really interesting question that's not fully answered.
Yeah.
I think we have a good sense of the kinds of molecules that clearly had to be around in the prebiotic Earth. Yeah. Where they came from is a question. I think this asteroid data is fascinating, right? Because it does kind of support the idea that Francis Crick, I think, first proposed, which is that life probably came from elsewhere, or at least the components came from elsewhere, because maybe there wasn't actually time for those molecules to evolve here on the planet, and or to emerge, you know, on the planet. Not known, but I think this new evidence is certainly interesting in terms of thinking about the origins of life. Maybe the components arrived here and were easy fodder for evolution to begin working on. Maybe.
Yeah. Just remarkable. And this idea, and then thinking about your own journey there, like from the prebiotic sort of chemical chaos, that was your PhD, due to this like highly precision-based editing and curing of individual diseases. Just, you know, it's been a remarkable journey.
It's been fun. I mean, I can't imagine having done anything else in a way, because it's been just such a wonderful career to be involved in the process of discovery.
The next 5 or 10 years, what do you think? We'll have our toolbox to use. We'll go recurrently to those.
The toolbox will continue to expand, that's for sure. You know, the capabilities will continue to evolve. That's also for sure. I think it's really a question of how to turn this kind of technology now into a turnkey approach to disease.
Yeah.
You know, whether we're talking about planetary disease or human disease, it's really about doing that in a way that becomes scalable. And I still think there's a really important role for academics and for nonprofits here, because I think it's pretty clear that, again, I defer to your expertise here on the clinical side, but humans are complicated.
Yeah. Definitely. Agreed.
Right? And, you know, this is why, you know, people always wonder, why do so many drugs fail in clinical trials? It's really hard. And I admire people that have devoted their careers to drug discovery, because it's a really hard problem. It's harder than anything I do, I think, you know. right? It's our work is the first step.
Yeah.
But then you got to actually show that it works in an actual disease situation and is safe for people to use, and that's a whole different ballgame. So I do think it's going to require continued, technological and scientific discovery to get us at least closer to that goal of being able to quickly come up with therapies that are effective for different types of disease.
I know a few drug discoverers who might argue that what you do is like easier than what they do. We're so happy to have you here. Thank you so much for spending some time with us. I can't think of few people who have impacted the world we currently live in more than you have. It's really just a pleasure to have the chance to chat.
Great to be here. Thanks again so much.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.